There we go. And I'd like to once again welcome you all to this seminar. And I would like to introduce our first speaker. Professor Makram Essafi is a professor of immunology at the Institut Pasteur de Tunis. And Makram is currently leading a group working on the role of innate responses to the establishment of an efficient and adaptive immune response against TB. And if you want to
hello
and start your
hello
sorry, are you there, Makram? Yeah, so if you'd like to start, thank you very much for this talk. You should just be able to share the slides now.
Sorry, I have to shut off my camera for better quality connection. So thank you very much, Blake.
Oh, we've lost your audio again. We can't hear you, Makram.
Okay, that's fine.
We can hear you now.
Can you hear me now?
We can hear you now, yes.
Okay, thank you very much. So I said thank you very much, Blake, and I would like to thank Validate for inviting me to give this talk on a part of our work that was about the procedure we were developed to enhance the efficacy of the parental BCG based on this transcription factor, the host transcription factor is called FOXO3. So I would like to start with this introductory about TB. You know, this tuberculosis is an airborne infection caused mainly by Mycobacterium tuberculosis. Top infectious killer in the world. Last decade, we had an average of 2 million people were dying by TB and 10 million were declared ill. Alternatively, Mycobacterium bovis can cause TB in human. That's the situation in Tunisia. In Tunisia, we have a particular situation. The incidence is not too high, but 65% they are extrapulmonary TB, then they are caused mainly by Mycobacterium bovis. And maybe one day we can give a talk about this particular situation. So also antimicrobial declared to have TB resistant form. But as I said, COVID-19 also has worsened the situation. Why? Because TB death that sources were dedicated for TB services, and the funds were mainly used to tackle COVID. Hopefully, the COVID-19, they were pretty successful, the vaccine to tackle this disease. However, for TB, we are still suffering and we do not have an effective TB. The sole vaccine used BCG is not that effective. TB made the situation more complex of many drug-resistant strains of TB, like we can talk about multi-drug resistance extensively and totally drug-resistant strain. Coughs or sneeze, and someone else inhales the expelled droplets which contain TB bacteria. So from here, there are three main scenarios. Majority will resist and contain the infection, develop what we call the latent form of tuberculosis of the TB. And there is about 2 billion people, they have this latent TB, and they represent a dangerous reservoir, you can say. Also exclude the fact that the possibility that certain persons get rid of the infection by their innate immune response. So starting from these, and the point that an effective immune response can control at least tuberculosis, drug-resistant forms with no use of antibiotics, along with the difficulty also to develop new vaccines better than the current BCG. Also working the development of what we call it host-directed procedure to better treat or and prevent tuberculosis. So this is our group. Talk about this work consists of explaining to you how we manage to correct the defect of BCG through targeting the host effectors that were behind the weak protection conferred by these vaccines. So BCG has been used for more than a century. A pretty interesting vaccine because it induces specific adaptive immune response against TB, which help, that means protecting disseminated against the disseminated and severe forms of TB, mainly in infants. What we call non-specific immune memory, that is the trained immunity. Countries mainly against other sepsis, respiratory infection, leprosis. However, BCG shows limited efficacy against pulmonary TB in adolescents and adults. Other BCG, we ask, what like others, how to make BCG better. Instead of developing another vaccine, can we make it better through what? So what are the main defects of BCG? There are mainly 3 defects. First, and this because for optimal presentation to the CD8 T cells, the mycobacteria need to lysosome get translocated to the cytosol, and the antigen will be processed and present to CD8 T cells. So BCG is enabled or it enabled to escape this phagolysosome. And this is why vaccine developed by the team of Kaufman. And this team, they made the recombinant BCG that expressed the stereo, the phagolysosome membrane, enabling the translocation of BCG to the cytosol, the processing of mycobacteria, and the optimal induction of CD8. What we call dendritic cell-mediated cross-presentation of CD8 T cells. So a apoptotic body charged with mycobacterial antigen will be.
Hi, Makram. We keep losing your we keep losing your voice. So are you able to get closer to the mic or anything?
Can you hear me now?
Yeah, I heard you perfectly then. So if you can do whatever you were just doing then, I should.
Well, when did I stop or did I have some problem? I can restart again from, okay. or just continue?
Just continue. Just continue.
Okay, thank you. So apoptotic bodies, as I said, charged with mycobacterial antigen will be ingested with dendritic cells. Dendritic cells are profession presenting cells. They will present the lipidic antigens and also through the class 1 to CD8 T cells. And it has been reported that there is direct correlation between the level of apoptosis of macrophage infected cells and between the adaptive immune response mediated by CD8 T cells. And this has been highlighted by many vaccine assays. For example, here, the same team of Kaufman, they were vaccinated mice with apoptotic bodies from BCG-infected macrophages, and they get higher protection. And the same team who developed this recombinant BCG expressing the stereolysin, this recombinant form of BCG induces high apoptosis, which may also explain the higher protection conferred by this form of BCG than the parental one. And also there is another team, they made this recombinant form of BCG expressing the human pro-apoptotic protein called BAX. And this form of BCG induces strong Th1 response in mice. Another feature of BCG, which is not a feature, but it's advantage, is that BCG induces a pretty high level of immunosuppressive cytokine IL-10. We know IL-10 is a negative regulator of immune response, and it blocks the development, the establishment of Th1 robust immune response. It inhibits macrophage-mediated killing of MTP, dendritic cell uptake. It inhibits also T cell polarization, as I said, for Th1. Even the migration of the Th1 cells from the lymph nodes back to the site of infection. In 2012, the team of Ogara, they used neutralizing antibody, monoclonal antibody. They were injecting this antibody during BCG vaccinated. And in this way, they blocked the IL-10 signaling, and they were able to enhance the immune response, the Th1, Th17. And they got in these mice high level of interferon gamma, TNF alpha, IL-17, and which led also to the higher protection against TB. So if we can characterize the cellular effector behind this apoptosis and controlling apoptosis, anti-secretion, BCG-infected macrophage, we may generate some tools, or let's say the adjuvant to augment the apoptosis and inhibit IL-10 during BCG vaccination, and we may enhance the efficacy of BCG. So in our group, we wanted to characterize or to look for these cellular effector controlling apoptosis and IL-10. And we chose to work on these transcription factors called FOXO, and we suspected the, we suspected the involvement of FOXO3 because this factor is considered as a tumor suppressor, because one way by which cancer cell, let's mean inhibit apoptosis, is to inhibit FOXO3. In the majority of cancer cell, this PI3 kinase AKT is activated, and the activation of this pathway would lead to the inhibition of FOXO3. While inhibition of this pathway by several known anti-cancer drugs, I think many anti-cancer drugs, they inhibit this pathway and they lead to the activation of FOXO3, which will induce the cell apoptosis through the induction of several pro-apoptotic effectors. And we also know that MTB has been reported to activate this pathway, PI3 kinase AKT pathway, to block the phagolysosome fusion. So this may be the mechanism used by pathogenic mycobacteria to inhibit macrophage apoptosis, like the case here, while the non-viral strains like BCG will inhibit this pathway, activating FOXO3 and leading to apoptosis of infected cells. So we used several cellular models to verify our hypothesis regarding the involvement of PI3 kinase AKT FOXO3 axis. And we used different MOI to infect cells in vitro. And we found that 24 hours after infection, we get a clear induction of apoptosis with high MOI. This high MOI mimic the vaccination that we use, because we use high MOI when we vaccinate people. So in order to also, that means to verify our hypothesis and follow the activity of FOXO3, we were checking the phosphorylation status of this transcription factor. And we found that 2 hours after BCG infected, this factor was totally dephosphorylated, which an indication of FOXO3 activation and translocation to the nucleus. Translocation to the nucleus was later verified by cellular fractionation. And as we can see here, at the steady state, FOXO3 is in the cytosol, but after infection, will migrate from the cytosol to the nucleus. And this migration was accentuated accentuated after using of PI3 kinase inhibitor. Translocation to the nucleus was further verified using fluorescent microscopy. So in order to assess also the effect of FOXO3, we knocked down the expression of FOXO3 in macrophage, and we found that when we knock down the FOXO3 expression, the apoptosis induced by BCG was diminished by about 30%, which is an indication of the direct involvement of FOXO3 in this apoptosis. Later on, we check it for the factor that are behind this apoptosis. FOXO3 is transcription factor, so we were looking for this effector of apoptosis. So we took the macrophages from 3 volunteers and we infected them with BCG, and we were looking and we did this small screening on 100 gene, and we end up by finding that 2 pro-apoptotic factors known to be controlled by FOXO3, they were induced after BCG infection. And these are Puma and Noxa. While the downregulation of FOXO3 with this function assay in macrophages was also associated by the downregulation of Puma and Noxa. We verified these also at the protein levels in human primary macrophages. Downregulation of FOXO3 also was associated with downregulation of Puma and Noxa. So the message here, we can summarize that BCG-induced apoptosis of macrophages passed through FOXO3 activation. Our study was also supported later on by at least 2 teams that they made a direct relation between FOXO3 expression. Actually, in TB patients that they have a defect in monocyte and macrophage apoptosis, they have low level of FOXO3 expression. So knowing that level of IL-10 in TB patient is pretty high, associating this with the level of FOXO3 expression that is really low, we asked, would FOXO3 be of IL-10 expression also? So we know the PI3 kinase AKT pathway is the pathway controlling in positive way the expression of IL-10. So we said this may pass through inhibition of the inhibitor. So if the AKT FOXO3 is the inhibitor of IL-10, inhibiting this FOXO3 by AKT will release the inhibition, get high secretion, and we get the secretion with the induction of IL-10. So to verify this, if we inhibit this pathway, so we'll release the inhibition of FOXO3 and we'll activate it in a way. And active FOXO3 will lead to the blocking abrogation of IL-10 secretion. So verified. That means the inhibition of the 2 pathways, diffuse-related FOXO3, activation of FOXO3, and the 2 cellular mode. Each time we add either the inhibitor of AKT by the MK or PI3 kinase by wortmannin, each time we were able to inhibit the IL-10 secretion induced by BCG, which is an indication that FOXO3 is an inhibitor of IL-10. We further verified the expression of IL-10, the regulation of IL-10 expression by FOXO3 using scRNA. So when we transfect cells, macrophages with the scRNA, the low FOXO3 expression was associated to higher expression of IL-10. And even the BCG-induced expression was increased when we knocked down the expression of FOXO3. The transcription regulation of IL-10 was also verified using gene regulation, gene reporter assay. So when we looked at the promoter of IL-10, we found 3 typical fork head motifs for FOXO binding, beside a typical FOXO3 motif. And when we transfected the cells with the promoter along either with the GFP or GFP FOXO3, we can see that the overexpression of FOXO3 inhibited the transcription activity of this promoter. Later on, we wanted to determine which of these motifs is responsible for this inhibition. So we mutated each site alone. And when we did our experiment, we found that this typical FOXO3 motif was the major responsible of this inhibition mediated by FOXO3 for the IL-10 expression. We also verified the direct binding of FOXO3 to the IL-10 promoter used by the sheep assay. Around to verify the effect of this regulation on macrophage differentiation. And we found when we downregulate FOXO3, we downregulate, that means the expression of M1 marker, while the overexpression of the activation of FOXO3 induce the expression of this marker. And most interesting point is that it makes these macrophages, when we activate FOXO3, more pro-inflammatory and enhance the bactericidal effect of these macrophages. The effect of this regulation also was assessed, and we wanted to check the effect of this regulation on IL-10 on adaptive immune response. So we took macrophages from a human one batch, and we divided on them in two batches. One batch was transfected with the cRNA for FOXO3. 2 days later, we added mycobacterial pathogen and co-incubated these macrophages with the autologous lymphocyte, let them 4 days to check the T-cell response. And what we found that when we downregulate the expression of FOXO3 in macrophages, with the adaptive immune response went toward Th2. That means that we get lower interferon gamma in absence of FOXO3 in macrophages, lower IL-17, higher IL-10 as expected, higher IL-4. This is mRNA level. And also the cytokine was the same. That means less interferon gamma, less TNF alpha, more IL-10, and less IL-6. So we can summarize that FOXO3 has the dual effect in macrophage infected with BCG. FOXO3 will induce apoptosis to induction of prima and noxa and also inhibit IL-10 expression. So here in presence of this pleiotropic FOXO transcription factor, as we said, mediate apoptosis through induction of prima and noxa, inhibit IL-10 expression, and also it enhanced, has been reported FOXO3 enhanced the survival of CD8 memory T cells. Also contribute to detoxification of gross radical oxygen species and also induce the autophagy. It's a host defense mechanism that is very important in the context of mycobacterial infection. And also FOXO3 induce antimicrobial peptide secretion. These are the reported genes. And we think that more function may be discovered of FOXO3 in the future. So if we push the activate FOXO3 macrophage, this would push the balance toward TH1, TH17, and we may make the better protection against MTB infection through induction of more apoptosis, low IL-10, induction of autophagy, and antimicrobial peptide. So would the activation of FOXO3, we ask would the activation of FOXO3 will lead to better, to enhance at least the efficacy of BCG. And for this, so we work on mice model through our collaboration with our collaborator in India. So we tested first in vitro the effect of this FOXO3 activator. We used this, well, it's an anti-cancer drug that has been developed by Merck. It's in phase 2 clinical study right now. And this factor has the ability to inhibit AKT in a very specific way and to enhance and also to activate FOXO3. And in vitro, we found also the inhibition using MK in vitro to inhibit the IL-10 expression, but also it induces, when mixed to the BCG, enhance the apoptosis induced by BCG. Each time we add MK, we get higher apoptosis in vitro. So later on, we assessed, that means the effect of MK in vivo. So these the 4 group of mice. We have naive mice. Mice were the other group of mice were vaccinated with BCG, the classical way. Mice injected with the MK, the activator FOXO3, and mice co-administered BCG plus MK. So 24 after BCG immunization, we injected the MK and the same site of BCG injection, MK, 24 hours later and 48 hours later. 4 days later, we collected the lymph nodes in order to assess the apoptosis of the whole cells and also on the antigen-presenting cells. And what we found, each time we add MK, these are the whole cells from the lymph nodes. MK induced and increased the apoptosis induced by BCG, either for the whole cells or for the antigen-presenting cells, as shown here in magenta. The effect of MK, that's on T cell response 3 weeks later. And what we found that MK induced monofunctional CD4 T cells. Addition of MK to BCG enhanced CD4 TNF alpha secreting cell, interferon gamma secreting cell, IL-17 also CD4 IL-17 secreting cell. The most important point is here we get polyfunctional, triple functional, these population of T cells that co-express TNF alpha, interferon gamma, and IL-17 when we add the MK, the activator of FOXO3. And we found the same data with the CD8 T cells. That means more CD8 T cells expressing TNF alpha, interferon gamma, IL-17, and these polyfunctional CD8 T cells, they express TNF alpha at the same time, interferon gamma, IL-17. Assess the effect of the addition of MK on the memory T cells. So we gated on the two T cell populations, CD4 T cells, CD8 T cells, and what we found that when we add MK, we enhance by like 50% of the central memory T cells, and also enhance the effector memory T cells in CD4, and also almost the same in the CD8 T cells. We end up assessing the effect of MK on the level of protection compared by BCG. So after challenge, the 30 days, 70 days later after challenge, we collected, that's mean the lungs, and we assessed it by CFU, the bacterial load in the lungs and the spleen. And what, as we can see here, the addition of MK enhanced the protection against MTB. 30 days, 75 days. We assessed these in two animal models, here in mice and here in guinea pigs. And as we can see here, even in guinea pigs, we get higher protection when we added the MK to BCG. So to summarize, we can say at the end of this presentation that BCG vaccination will induce higher IL-10, which is immune suppressor, and we get less apoptosis for antigen processing, less efferocytosis. So the cross priming will not be optimal, while the activation of PI3, the activation through MK, that means the inhibition of this pathway, will inhibit IL-10 secretion and increase the apoptosis. Get more apoptosis, more antigen processing, low IL-10, more efferocytosis. That's more apoptosis, more cross-priming, and activation CD8 T cell, leading to an effective, robust immune response. Excuse me, there is, I don't know why it's... Sorry. Yes.
Are you okay? Are you okay to wrap up on the... Sorry.
Yeah, I finished. So by the end, so yeah. And now in our group, we are thinking about developing maybe host-directed therapy procedure through the activation of FOXO3. And what encouraged us really, this point here for this figure from the vaccine study, the sole use of MK for 2 injections was able to confer some protection to these mice. And also in macrophages, when we added the MK, the activator of FOXO3, enhanced the bactericidal effect of macrophages almost in the same way as isoniazid, the antibiotic used to treat TB. So, and now we are looking for collaboration if you want to perform this study. So finally, I would like to thank my student, Maryam Ahwas, PhD. She started the work on the involvement of FOXO3 in macrophage apoptosis. And she started also the work on the regulation of IL-10 by FOXO3. Rania took over, she continued to work on IL-10. And after that, she went to the lab of our collaborator, Aman, that I would like to thank, where she performed the vaccine study on mice. Amira also contributed to both works. I would like to also to thank our institution, our ministry, and World Health Organization, WHO, who gave me this the starting re-entry grant to try and to start the work on the involvement of FOXO3 on TB immune response. All guys, and you are welcome to visit Tunisia.